recording at the end what I started this recording off, what I started off saying to you guys before the recording got turned on. Okay? So you'll go to the end to review the phases of oogenesis and the creation of a mature follicle and a secondary oocyte, because that's what's in a mature follicle. And then you'll go ahead and take a look at the basics of the hormones and the endometrial lining. So let's just take a look at these charts here, because we can kind of summarize some of the stuff in these charts. I'll erase a few things I put in. Okay. Ovarian cycle. What the top chart is meant to show you is this are the visual changes that you can see that's occurring in the ovary. And so what we are looking at here is ovary and follicles. Remember, when ovulation occurs, this is a secondary oocyte. A primary follicle is a primary oocyte. And all of those cells, all those granulosa cells, are responsible for making hormone estrogen. They produce estrogen. And so the peak estrogen levels that you see right down here are all due to that mature follicle. Now, the follicles started to develop. A bunch of follicles were recruited to become what's the primary follicle. And remember, follicle is what we see in the ovary, okay? They were recruited to become primary follicles, which means we go from simple squamous epithelium to cuboidal epithelium to producing hormone. So let's say right here, we're going to get a group of approximately 20, just because it's reasonable, to start this race. And this one is the winner, which means the others are losers and they die in atrophy. But that is what's going to be, all these groups are going to be what's contributing to this rise in estrogen. So that's what FSH is for, follicle stimulating hormone. That's the one that says, okay, let's start the race. And it's low estrogen levels which will allow FSH and LH to be released. So low hormones, E and P, by the way, not just estrogen, but low estrogen and progesterone levels. These low will cause an increase in GnRH, and GnRH will cause FSH and LH to be released. Ovulation occurs because now E stimulates this, which stimulates ovulation. Then corpus luteum is a P, progesterone producing, primarily, it's still producing estrogen as you take a look at things, unit, and is responsible for creating the secretory endometrial lining of stratum functionalis. E is responsible for all of this proliferative phase. So it helps it build up, and then progesterone helps it get ready for implantation, so it's lush. And if implantation of a fertilized egg does not occur, then we don't have human chorionic gonadotropin released, HCG released, and we don't have maintenance of the corpus luteum, and we lose the lining. So now we can go into this part of the notes. Follicular phase is days 1 through 13. So we start off with our primordial follicles. which remember are containing primary oocytes. And say we're going to recruit a group of around 20, just because it's a nice thing to continue with. And we're going to ask them, because of GnRH and FSH and therefore LH, we're going to ask them to become a primary follicle, which still contains primary oocytes. Primary follicles will then turn into a single secondary follicle. And that's why I have S. They all want to become primary follicles, which is layers and layers of granulosa cells without little spaces in them. When you become a secondary follicle of that group of 20, that means you're going to become the winner of this race, and you will then become the vesicular or mature follicle. Ugh. If you are a mature follicle, right before you're ovulated, you become the egg in the middle, the oocyte, becomes a secondary oocyte. And remember, secondary oocyte goes into meiosis II and gets stuck in meiosis II. So GnRH causes FSH and LH release. FSH is follicle growth. And will cause follicle development, which I just talked about right up above, in that sentence up above. Primordials will become primary, primaries will become secondary, secondaries will become mature or vesicular follicles. And it is the granulosa cells, PD, produce estrogen. As estrogens increase, we'll have negative feedback on FSH. Because FSH is for follicle development. We don't want any more follicles developing. We have one that's developing, and this is the winner. We don't want another race to go on at the same time. And so FSH levels drop. Until we have this critical level here of estrogen. And that's coming from the mature follicle. That critical level of estrogen is coming. So now, suddenly, we are positive feedback on FSH and LH, but it's just it causes a surge. And the one that we are most important, what we want to pay attention to is the LH surge. That's the one that causes ovulation. In ovulation, the actual wall of the ovary ruptures. And the oocyte is now expelled into the peritoneal cavity. Now, is this a primary oocyte or a secondary oocyte? Secondary. Whenever ovulation occurs, we're releasing now a secondary oocyte. So make sure it's complicated, you know, and everybody says women are complicated, right? So there is some rationale to this, even at the germ cell level. You know, most women don't think they're complicated, but other people that you talk to think they are. You guys know who I'm talking about. Oh, come on. If you can't joke a little bit about some of this stuff, you're even more serious than I am, which is really pretty bad. Okay. Now, let's go into the luteal phase, and that's day 15 through, that's supposed to be an arrow. I don't know why it does that sometimes. Through day 28. And remember, this 28-day cycle is a perfectly cycling female, and not all women are perfect cyclers. So not every cycle is going to occur every 28 days. But what's important is the luteal phase. In a woman that has irregular cycles, this is the one you can bet on, is consistent. Once ovulation occurs, you don't know how long the preceding stage is. You don't know how long the follicular phase is for women who are irregular cycles. But women who, once they do ovulate, this luteal phase is the most consistent, no matter what her cycle's length is. So. And the reason it's most consistent is that now the ruptured follicle collapses and becomes the corpus luteum, and I'm going to abbreviate it CL, corpus luteum. The CL then changes H production and becomes progesterone, I'm going to spell it out, progesterone dominant. And you can see that from the graph or the chart from up above. So after ovulation, progesterone levels rise to a peak. Estrogen levels rise too, but the dominant hormone now being produced is progesterone. One of the things that's important for you to realize is that this is important to create a healthy uterine lining for fertilized egg implantation. The other thing too is high P levels all by themselves have a negative feedback on GnRH, FSH, and LH production and release. Because if you take a look at up above where we have all that, again, this area of the cycle, the secretory phase, look at that. And then look how when they drop, all of a sudden, look what happens to FSH and LH hormone production. And their drop also is consistent then with the lack of support for that, what we call secretory endometrium, which is part of that stratum functionalis. So if there is no fertilization of the secondary oocyte, what happens is CL dies. It disintegrates, it degenerates, whatever you want to say, and becomes CA, corpus albicans, which means P levels drop, which then means next menses. And I emphasize progesterone because that's the hormone that is totally responsible for the maintenance of the secretory endometrium. Estrogen primed the pump, but progesterone keeps it there. So without progesterone, you lose that secretory endometrium. Yes?
What does a triangle mean on the first row?
Oh, change. Delta. Delta's change. Good question. I have all kinds of shorthand ways that I'm trying to teach you. And some of them you like and some of them you don't like. Okay, so if fertilized, that means implantation necessary for maintaining CL, the corpus luteum. So fertilization and then successful implantation must occur to keep that corpus luteum, because then the conceptus releases HCG, human chorionic gonadotropin. That's what actually is measured in those urine pregnancy tests, that level. And it's only released by a successfully implanted zygote or conceptus. When it releases HCG, this tells CL to live for 3 more months. So corpus luteum will live for 3 more months, which means what about hormone levels? They stay high. So the corpus luteum is responsible for producing progesterone predominantly, but it still produces estrogen, and progesterone is absolutely essential for maintenance of the uterine lining. And so when a miscarriage happens, you know something right off the bat, right? What hormone is deficient when a miscarriage happens? Progesterone. So when progesterone deficiency occurs during that 3-month time period, if for some reason during that 3-month time period, and isn't that when most women actually have a spontaneous miscarriage? During that 3-month time period, that means something happened to the progesterone levels. And when progesterone levels drop, the uterine lining is not maintained, and so therefore that causes either the conceptus died beforehand or the conceptus will die because of. And you don't know. You know, we don't actually take products of conception and analyze what came first, because actually it's really, really hard to tell because they actually happen so close to one another. But if conceptus dies, then the corpus luteum will no longer receive signal of HCG and it will die, and then sloughing will occur. But if for some reason the conceptus does not produce enough HCG, then the corpus luteum will no longer get its signal to survive, and then it will, progesterone levels drop and sloughing occurs. So it's really, really hard to tell on this one which happens first. Typically, I will tell you, typically there's something wrong with the conceptus, and so that leads to then the snowball effect where this happens. But that's why that progesterone level is really, really super important, and the corpus luteum is where progesterone comes from in the first trimester. After that, when we get into the next chapter, you'll find out that the placenta takes over. It's mature enough at that stage and actually is responsible for, because that's one of the things I want you guys to realize, the maintenance of the uterine lining is a progesterone process. So throughout the pregnancy, progesterone levels are high because that is what maintains the uterine lining. So menstrual cycle, 3 phases in sequence at the same time, occurring in the uterus and because of what's happening in the ovary. Because the menstrual cycle is hormone dependent. Uterine lining is what the menstrual cycle is all about. Uterine lining is H dependent. And so it has everything to do with estrogen and progesterone levels. So before puberty, what do you think the uterine lining looks like inside a uterus? Pretty skimpy, okay? Because the uterine lining builds up, the stratum functionalis builds up because of estrogen and progesterone. Stratum basalis is always there, but stratum functionalis is kind of like non-functional. It's not really there until puberty hits, when hormone levels actually start to fluctuate. So in the menstrual phase, which is day 1 through day 6, or day 5 or 6, that's what's called the shedding phase. And this is when E and P at lowest. So that means GnRH goes up, which means FSH and LH go up. Now, FSH and LH go up because estrogen and progesterone levels are low. Once we start follicles developing, what do we know about estrogen levels when follicles start to develop? Estrogen levels start to go up, and so that's what the proliferative phase is all about. It's the rebuilding because we just sloughed. And so now E levels increasing more than menstrual phase. And why do I say that there's any E levels in menstruation? E starts exceedingly low during menstruation, but because now follicles are starting to develop in the ovary, E levels start to go up. So in the proliferative phase, around day 6 to day 14, E levels are definitely increasing. Then the secretory phase occurs this is all uterine lining, occurs after ovulation. So for day 14, ovulation. Now we have day 15 through 28, preparing the uterine lining for the embryo. And so this is due to increased P levels. And of course, increased P levels are because of corpus luteum activity. And again, if no HCG to send message to Cl, then Cl becomes Ca, corpus albicans. And so this is typically a 2-week time frame, which is day 15 through 28. No HCG, menstruation. HCG, maintenance of the uterine lining and the first trimester. Female sexual response. So the physiological events that occur during sexual encounter with excitement and orgasm, and then the resolution phase. What's important is testosterone is the sex drive hormone for men. Well, for women, it's not testosterone, it's DHEA, dehydroepiandrosterone, and it's an androgen. So DHEA is actually the hormone that's responsible for the sex drive in the female. With excitement phase, this is going to be analogous to the male erection phase. This is going to be due to parasympathetic stimulation plus sympathetic. The parasympathetic nervous system is going to actually cause vascular engorgement of secondary sex characteristics and organs, such as breasts, clitoris, vaginal wall, and labia. That's all under parasympathetic control. So this is interesting. This is most vascular structures, blood vessels in other words, are under sympathetic control. With our genitalia, that's the only area that is not under sympathetic control. It's under parasympathetic control when stimulation and excitement actually occurs. Now, the sympathetic things that we'll actually see is increased heart rate, increased BP, blood pressure, and increased respiratory rate. Those are all due to the sympathetic system activation. When orgasm occurs, that means we actually have a peak in the activity of the parasympathetic system actually occurring, and then the female will experience pleasure and have release of tension that's building up during this time, and actual contraction of the uterus and vaginal wall. Smooth muscle contraction will occur. And this phase is dominated by the sympathetic nervous system. The resolution phase is the reverse of vascular engorgement. So basically, all of these areas that became vascularly engorged will now actually allow the blood to actually flow out. So that's the female system. Now we're on to the men. Don't sound so ready. Got done talking about gals, now we'll talk about guys. All right, so here we go. Anatomy of the male reproductive system. Now we'll start externally with the scrotum, which is a pouch of skin that houses the testes. And a mature individual will actually have hair over that scrotum itself. The scrotum itself is designed to actually keep the testes at a temperature that is lower than body temperature. And the reason we need to do this is that for men, this is actually necessary for sperm cell production. So in order to make sperm cells that can fertilize an egg, the testes need to be outside of the body cavity, because they actually originated inside the body cavity. They need to be outside the body cavity to where it's cooler, and that's what the scrotum is for. Keeping them close by, but not too far away, because we have to actually have some body temperature. It's just a couple degrees cooler than inside. So if somebody has an undescended testicle, what does that mean? It's inside the body cavity. What do you know about the fertility of that testicle? Not fertile, okay? Now, if you have an undescended testicle, they can easily go in and surgically help it get into the scrotum itself, and then that testes will then have the ability to actually produce fertile sperm cells. So that's important, that it actually is outside the body cavity at a lower temperature. We have two muscles that help to control the location of the testes inside the scrotum. And that is the cremaster muscle, which is only around the testes. And the other muscle is the dartos muscle, which is inside the scrotal wall. So the cremaster is around the testicle, and the dartos muscle is actually part of the scrotal wall. And so when you contract either one, what do you suppose will be the consequence of contracting either one? Pulls the testes up close to the perineum or the body wall. Okay, so they both function. And why do you suppose we have two systems here? Yeah, there's always a backup. So either one can do this for us. But typically, in a normal, healthy male, both will function, but either one can do this. And so why would you bring the testes up close to the body wall? It's cold outside, absolutely. And so when it's hot, they're actually further away from the body wall. When it's cold, they're up closer to the body wall. That's just a normal physiological event because the body wants to actually be able to produce normal functioning sperm cells. So those are the function of those two muscles. One's around the testes and the other is actually part of the scrotum itself. Testes are the male gonads or primary sex organs because they produce hormones, which is testosterone, and gametes. Which we are going to call spermatozoa. As you are familiar with the cat anatomy, the testes has a couple of tunics. Tunica albuginea is one I prefer to talk about first because that's the true outer limiting border of the testes. So it's the fibrous CT coat that's inside the tunica vaginalis. But it is actually the border and will have septations, which is extensions of that CT going into the testes to actually subdivide it into lobules. Of which there are about 300 per testy. The tunica vaginalis is actually the outer layer that comes from the peritoneum. And this is, we'll stop there with this description. And the reason that I like to talk about this close to actually the need to be outside the body wall is that the testes start in the same general area that the ovaries do, their development does. So they're intra-abdominal. So they're intra-abdominal organs that actually start to descend because where you guys find the testicular blood vessels, that's where they were. Where you find them coming off of the IVC and the aorta, that's where they were. So they actually take a descent, and those blood vessels are very happy to actually stretch and stretch, and they go all the way down to what's called the inguinal canal. When they hit the inguinal canal, remember, they have a cover over them called peritoneum, correct? Anything that's in the peritoneal cavity has a peritoneal covering. It's visceral peritoneum. Well, once they hit the inguinal canal, they actually then take the parietal peritoneum that's covering up the inguinal canal. If you guys can envision this, there's an internal opening, which means inside, and an external opening, which means outside. So they go into the internal opening of the inguinal canal and take parietal peritoneum with them, go down the inguinal canal, and then come out the external inguinal opening, and still have that peritoneum over them, and that's tunica vaginalis. So you see it kind of, they have their own visceral peritoneum, but then they actually push through the parietal peritoneum, and that's what you guys see. That's part of the spermatic cord and also makes up the external aspect of the tunica vaginalis. I'll stop there, and we'll probably finish this chapter almost finish this chapter next time. So I'll put a pause on the recording. Okay, this is actually the beginning of my lecture, which wasn't recorded because I was having problems with my symposium allowing me to write on it. So let me go back and again reiterate some of the things that's important for you to get a good grasp on in order to understand the ovarian cycle and the other things that accompany it. Remember, oogonia are the female's version of germ cells of the gonad. And what happens is when you hear oogonia or spermatogonia, I want you to realize that these cells have 2 sets of chromosomes and are the cell line that undergoes mitosis. So mitosis means from one mother cell you'll get 2 daughter cells which will resemble exactly the mother cell. And so our oogonia of the female ovary during embryology and fetal period will be very mitotically active, creating more and more diploid cells, which means 2 sets of chromosomes. And once the ovarian population gets established to several million, then the oogonia will stop their mitotic process, and then suddenly, boom, they actually go into meiosis 1. When they go into meiosis 1, they become primary oocytes. So a primary oocyte tells you right off the bat, this is now an egg cell of the ovary that doesn't go into mitosis anymore. It's all meiosis. And meiosis 1 is called reduction division. And what that means is that you will go from 2 sets of chromosomes to 1 set of chromosomes. And that means that your cell will go from diploid to haploid. So meiosis has 2 stages. It has meiosis 1 and meiosis 2. In meiosis 1, a primary oocyte enters and gets stuck in prophase 1 of meiosis. And that is where that oocyte stays until it is puberty. Once in puberty, then a whole bunch of these oocytes that are stuck in meiosis 1 and therefore primary oocytes will become recruited by gonadotropin releasing hormone, which will cause the release of FSH. So the true stimulus is FSH, follicle stimulating hormone. So follicle stimulating hormone acts like a recruiter, and it comes up to a group of primary oocytes that are primordial follicles and says to a group of, say, 20, I would like you to actually mature, or actually, it says a little bit more silly story, I would like this group of 20 of you to enter this race and complete this race of meiosis 1 and become the winner, which is the secondary oocyte. When you are the secondary oocyte, that means that you will have gone from a primordial follicle to a primary follicle, secondary follicle, mature follicle, and you will be ovulated. When you are a mature follicle, then what that also tells you is that you won the race. All those other competitors of this group of 20, in other words 19, will have failed and will not be able to keep up with you, and because they cannot keep up with you, they will atrophy and die. So they may start out, everybody starts out primordial follicles and all become recruited to become primary follicles, but the one that develops the fastest, which actually develops this increased layering of the granulosa cells, and therefore with increased layering of granulosa cells, increased estrogen production, which as the estrogen production increases, let's go over here, down here, as this estrogen production level increases, it will have negative feedback on FSH. So see how that FSH line dips down. Well, you need FSH for follicles to develop or to be recruited. And so that follicle that grows the best and produces the most estrogen will then cause FSH levels to drop, which will then cause the other follicles, which aren't producing that much estrogen and stimulating the whole process, to then actually atrophy, because in order for these follicles, other follicles that are kind of lagging behind to win the race, they need to be recruited and encouraged by FSH to continue their development. So FSH levels drop because estrogen levels are rising and have therefore a negative feedback on FSH levels. So when you see a secondary follicle, remember it has fluid-filled areas called antrons in it, and it has a whole bunch of granulosa cells, and that's essentially the winner. That secondary follicle, there might be a few others, but typically with normal ovulation and producing a ovum for fertilization, there's typically only one, unless of course there's a double ovulator, which means that there was basically a tie in this race, and so there were two winners during this race. But this secondary follicle will go on to become a mature follicle, and when it's a mature follicle, you are now turning into a secondary oocyte, which means you have completed meiosis 1. So secondary oocyte is developing that mature follicle, and for sure, even if you're not quite sure, a secondary oocyte that's ovulated, it is always, when you've ovulated, you are now a secondary oocyte. So a secondary oocyte means I've completed meiosis 1, and I've created that big lush cell, that's the egg, and that little polar body. Okay, this is the reduction division. Both are haploid, but that little bitty polar body doesn't have the lush cytoplasm that we need for that 2-week-long survival trip to go down the fallopian tubes and successfully implant into the uterus. So that's what meiosis 1 and 2 is designed to do in the women's system. It is designed to produce an egg that has copious amount of cytoplasm and mitochondria and all the cellular machinery needed for survival. Now, when that secondary oocyte leaves the ovary during ovulation, that surrounding granulosa cell layer, which was producing all that lovely estrogen, now gets its machinery changed. And so when you take a look down here at our estrogen and progesterone levels that are coming from the ovary now, estrogen and progesterone is coming from the ovary, we see that estrogen levels after ovulation drop because the cells are changing their primary hormone machinery production. And now they're switching from estrogen dominance to progesterone dominance. And again, estrogen levels will again be produced, but it is this progesterone. Progesterone is the big player. And when we take a look down at the uterine lining, where it says uterine cycle, progesterone is the absolute hormone that's absolutely necessary for creating that secretory phase. Now, the secretory phase is like making the uterine lining like the best petri dish ever for survival of a fertilized egg. And so we need that progesterone in order to do that. But in order to recreate a normal stratum functionalis, that's what the proliferative phase is all about. So we need that estrogen to give us the estrogen hormone to help us rebuild the functional layer of endometrium that was lost during the menstrual phase. So the menstrual phase occurs because E and P are at their lowest levels. And so then we get menstruation. E, P, low, low, low. And because of that, we have no maintenance of stratum functionalis, and then we get menses, which is, depending upon the individual, 5 or 6 day time frame. Then, because our E and P are at the lowest levels, then guess what happens to gonadotropin releasing hormone? That stimulates it to be released, and when gonadotropin releasing hormone is released, then FSH and LH is released. And when FSH, more importantly right now, FSH is released, follicles start to develop. So we'll start off with very low E and P levels at the beginning of menstruation, but because now we're recruiting some follicles in, look what happens to our estrogen levels. They start to slowly rise. And then as one of our secondary oocytes, or secondary follicle is getting large enough, produce enough estrogen hormone, we get this peak, and the mature follicle is the prime producer of that sharply rising estrogen levels, which is absolutely necessary. This peak estrogen level here is absolutely necessary to create this LH surge. So this LH surge is actually what happens, or is important, it has to happen in order for actual ovulation to occur. So that's the cycle. Remember, the last 2 weeks of the cycle are the most consistent as far as hormone levels, and women who have irregular cycles what happens is it is the proliferative phase that's actually quite variable for them. So they have strange gonadotropin releasing hormone levels, FSH and LH actually occur. So I think that summed up what we missed out from the beginning of lecture. Do please note there is a very good 228 forum posting about this, and I hope that you go ahead and review that, and if you have any further questions that need clarifying, please add to that post. How'd I do? All right.